Hello everyone, today I will talk about my research related to the computational design and modeling of biological systems. It is about using a model-driven engineering approach for synthetic biology using a framework called virtual parts. I will mention some of the new features and how computational tools can use this framework. Finally, I will demonstrate the computational modeling of an existing biological design using this framework. We all know that biological systems can be difficult to engineer. These systems can be really complex. Biological systems are also noisy and multidimensional. Another challenge is that these systems can have huge design spaces. There may be several solutions even when we use only a small number of parts. The design process in synthetic biology starts with identifying some biological parts that may be used. We then somehow use these parts to design desirable solutions. When we say a solution, we actually mean some biological behavior or function. For example, we may want to design a genetic circuit to sense some environmental signals and then respond by producing some proteins. The resulting proteins can be perhaps quantified in a lab environment. It can be very challenging to start with a list of biological parts and then to find out final DNA sequences encoding our desired solutions. One way to create this mapping is perhaps to use computational models. We typically integrate the individual behavior of biological parts by writing some mathematical equations. We can also incorporate information about how these parts interact with other parts, again in the form of mathematical equations. The resulting models can be simulated. That's great. This allows us to predict the behavior of our resulting genetic circuits. Hopefully, we may be able to narrow down some of our solutions to derive DNA sequences that can encode our desired systems. This approach is really useful, but it's not scalable. As the number of parts increases, we will really need to incorporate a lot more information into these models. The complexity may then increase exponentially. Clearly, manually creating a model for every alternative solution is not feasible. One way to automate this approach is using a model-driven engineering approach. We developed a method to provide modular models of biological parts. And these modular models can be combined computationally to derive models of desired systems. Essentially, each resulting model represents a solution. Engineering design process may include design-build test cycles, and there are also various other steps. First of all, the virtual parts framework that we developed integrates data from several data sources. These data may be part-specific, such as types and biological roles of parts, and also other constraints. For example, a part may be functional in the presence of another, or a DNA-based part may have some contextual dependencies. Our framework integrates related data and makes it available to computational tools, which can then query the underlying repository to start building designs. The virtual parts framework provides models of these parts and interactions. As you will see later, these models can be joined together either by tools or by using the methods provided by the framework. Parameters can also be changed by tools. The resulting models are in standard formats actually, and they can be simulated by off-the-shelf simulators. These computation simulations will facilitate choosing an alternative solution. We can evolve the circuits by tweaking different parameters or adding or removing parts, which will then change the overall behavior of a circuit. As I mentioned, over the years we developed standard virtual parts. These are essentially a mapping between genetic parts and their mathematical models. These models are modular, they are reusable and composable. These properties facilitate providing a model-driven engineering approach. Information in these models is also reused via templates. Each model derives from one or more templates, which can also refer to other templates. Each model can incorporate several different modeling entities, and they are defined with some inputs and outputs. These inputs and outputs make the models composable. These models are created based on a modeling abstraction, obviously. Recently, we introduced the use of rule-based biochemical reactions. In this approach, biochemical reactions are implemented based on binding and unbinding of biological molecules, and simple rules are aggregated to represent complex relationships for scalability reasons. This also makes the mapping of usage requirements to complex models easier. The modeling abstraction that we use is at the level of individual DNA sequence fragments, proteins, small molecules, and also the specific interactions between them. Here, for example, we have 4 different biological components at the DNA level, and we use these parts to produce some proteins. Over time, there will be several instances of this protein. The arrow-like components here are called promoters, and they initiate protein production. Sometimes circuits will include several other components for multiple production units. And sometimes these production units may be a bit more complex, such as having multiple promoters. The production rate in these cases may be different based on the availability of these promoter components. They may become unavailable when they are occupied, and the complexity may increase further due to the properties of different components. For example, these promoters may have additional binding boxes around them for different molecules to bind. As a result, the availability of these promoters and whether they are active or not will be based on a temporal function. There are also other desirable features. We may want to deploy different parts of a circuit using plasmids, which will then allow us to control copy numbers. We should also incorporate environmental signals and contextual dependencies. We recently improved our framework to facilitate design-to-model conversion. This conversion allows us to separate design and modeling layers for computational workflows. The framework now accepts designs in a standard language called SBOL and can create corresponding models in the form of SBML models. I'll come back to this. As a result, design tools can take advantage of our framework. SBOL, or the Synthetic Biology Open Language, allows specifying genetic circuits hierarchically. Here we have a receiver component and also a reporter component. The receiver component has some biological parts to produce 2 different proteins. The first one converts an environmental signal into a modified form of the second protein. This modified protein can then activate the reporter component to produce some green fluorescent proteins that can be quantified or measured in the lab. This receiver and the reporter parts can be defined as modules, and they can be connected using SBOL. The SBOL language also allows us to give coordinates by actual start and end locations of DNA-based components, or we can use relative constraints. For example, a component may precede or overlap with another. The models are created in the form of SBML, or the Systems Biology Markup Language models. As I mentioned, the resulting models may include submodels hierarchically. The framework has a modular architecture. It has a graph-based backend. Although this is the default database, tools can also use other compliant repositories. For example, the VPR data layer has been adopted by SymBioHub repositories. Therefore, SymBioHub repositories can also be used. These remote repositories allow designs to store, retrieve, and share their designs using SBOL language. Tools can use our framework in 3 different ways. First of all, in the connected mode. In the connected mode, tools may have some information about how these DNA parts are ordered. As a result, the VPR will return a complete model to simulate the behavior of this circuit. As you can see, VPR integrates additional information from the repositories when creating these models. In the disconnected mode, designers may have all the information. Here we have the genetic production interaction and the transcriptional inhibition interaction already added by the designer. VPR can be used as a design-to-model converter without any interaction to any of these databases because all the information is already included. If the design doesn't specify any biochemical parameters, then VPR will provide default or nominal values. Thirdly, the VPR can be used to enrich existing designs using the data mode. This is quite useful for tools that already have their own modeling abstraction. Tools can submit their designs and VPR enriches those designs with additional information by including constraints from the selected repository. We also provide different approaches to retrieve data or models. First of all, information about parts or models can be accessed using the VPR client library. It's currently available in the Java language. You can submit an SBOL design or the order of parts, and you can retrieve a model as a Java object. Another programmatic access without relying on a particular programming language is via the web service. VPR comes with a RESTful web service, and models are returned via HTTP. This makes the command line access quite easy as well. You can use off-the-shelf tools such as curl. You can easily post parameters, and the following lines here actually are for the model returned by the repository. In addition to the API and the web service access, the repositories can also be queried directly actually. Data are stored as fragments of graphs anyway. This means we can use a graph pattern language like Sparkle to retrieve information from a VPR-compliant repository. Let's look into a toggle switch example, which has been frequently used in synthetic biology examples. In this toggle switch system, when we set the input, the output becomes 1. Alternatively, we can reset the input to 0 and the output will become 0. In our system, in the presence of ATC molecules, the system produces RFP, red fluorescent protein molecules. If we switch the input to IPTG, the system will produce GFP, green fluorescent proteins. So ATC for RFP and IPTG for GFP. When ATC is present, it deactivates the inhibitor of RFP molecules. As a result, the system will produce RFP, but at the same time, it will produce LockI molecules that will inhibit the production of GFP. Similarly, when IPTG is present, it will lead to GFP production and also TET-R production, and TET-R acts as inhibitor of RFP production. In a Nature Communications paper, a real-time monitoring of this system was presented in 2017. Both IPTG and ATC signals were kept in the system for a specified period, and they were washed away at the end. The length of these periods define how the system will behave and whether to favor the production of GFP or the RFP molecules. ATC is kept for 7.5 hours, and then the system provides IPTG only signal for another 7.5 hours. The system oscillates between RFP and GFP production states. As you can see, due to the properties of parts used, RFP is produced more compared to GFP. To simulate this system, we first selected parts that can be used to build this circuit and arrange them in the same order. Our work includes computational design and simulation only here. Before starting the simulations, we used data from another study from the Void Lab. We used the reported expression values by Nielsen et al. And we parameterized our models. As you can see, we kept the input using the similar intervals. We could somehow show similar behavior regarding the RFP and GFP proteins productions. If IPTG is kept for 2 hours and if ATC signal is provided only for half an hour, then the RFP to GFP ratio oscillates around 1. However, later on, the system favors the production of RFP when both inputs are present in the system. We then wanted to simulate this behavior by only changing the intervals of IPTG and ATC inputs and also the final amounts of these inputs. In fact, after 15 hours of simulation time, our model also observed a similar behavior, favoring the RFP production. We were able to show the reusability of biological models that are parameterized with data from one study to simulate a design from another study. The documentation about how to use the client API or the web service via HTTP or from the command line is available via a wiki page. Thank you for listening, and special thanks to my collaborators, colleagues, and the SQL community, and also to everyone who contributed. If you are interested in using this framework, please get in touch with us. You can read more about this work in our recent ACS synthetic biology paper. Thank you.